during question time. So let's get things started. So Dr. Wang graduated from Peking University Health Science Centre in 2004. She then undertook her PhD at the University of Rochester and started working on cardiovascular disease. After graduating from her PhD in 2009, she joined Brigham and Women's Hospital at Harvard Medical School as a postdoctoral researcher. And in 2011, she was awarded an investigator of the American Heart Association and promoted to an instructor. In 2013, Dr. Wang won the Harvard Chinese Outstanding Young Investigator Award and was selected for the National Young Talent Program. And in 2014, she returned to China and was appointed professor at Peking Union Medical College. She is the Associate Director of the Department of Pathophysiology and also Associate Director of the Institute of Basic Medicine. And her research focuses on the pathological mechanisms and molecular diagnosis and therapy of cardiovascular diseases. So thank you very much for joining us today. Really looking forward to hearing about your paper and congratulations on being selected as the editor's choice for JMCC.
Sure, thank you. Is that okay? Let's start.
Perfect.
Okay.
Thank you.
Okay. First, thanks for introduction from Dr. Xiao as the host. Coming from Peking University. And first, I want his appreciation to show my great appreciation to JMCC and ISHR committee for giving me this opportunity to share our recent work about the role of microinflammatory in cardio remodeling. So what I will present today is about the microinflammatory contribution cardio remodeling by promoting cardiac hypertrophy, fibrosis, and inflammation. For heart failure, the inability of the heart to provide sufficient blood to the body remains one of the leading. Pathological cardio remodeling plays an important role in the development of the heart failure and is characterized by the finally leading to the functional defects. The prevalence of the definition and region, but has been estimated 1 to 2% in developed countries. Mechanisms of the heart failure include nervous system, activation of RS system, cardio remodeling, and halo factors, including AMP and BMP, and some pro-inflammatory cytokines such as L6, L1 beta, TNF-alpha, and IL-18. The therapeutic drugs for heart failure at present mainly include beta blocker, RS inhibitor, such as ACE inhibitor and AT1 receptor blocker, and diuretics and positive inotropic drugs. Recently, it has been demonstrated that inflammation plays an important role in the heart failure. Risk factors for heart activation of immune system, increased infiltration of pro-inflammatory cells, including monocytes, B cells, and T cells. And pro-inflammatory cytokines will activate cardiofibroblasts, which result in the excessive production of the extracellular matrix. Related production of TNF-alpha and L1 beta in blood mononuclear cells, where activation of NF-kappa B microin could play a role in the cardio remodeling. Which consists of alpha and beta subunits, zinc adopeptidase, and are widely expressed in intestines as well as in some cancer. So maprins, what is maprins? Maprin, which consists of alpha and beta subunits, belongs to taxane family of the zinc adopeptidase and are widely expressed across mammalian kidney, intestines, as well as in some cancer and immune cells. A major difference between both enzymes is so-called inserted domain in maprin alpha, which include a foreign cleavage site, This results in the loss of the EGF-like transmembrane and systolic domains to subsequent release of the enzyme into the extracellular environment. Besides, in skin tissue, miprin alpha was reported to remove C and C-terminal and N-terminal propeptides of the pro-collagen 1 and collagen 3, thereby releasing mature collagen 1 and collagen 3, suggesting the role of miprin 1 alpha in fibrosis. So based on our previous introduction, miprin 1 alpha may play a role in inflammation and extracellular matrix remodeling. So we are wondering whether miprin 1 alpha could mediate cardiac remodeling and heart failure. So to further explore the role of miprin 1 alpha in heart failure and cardiac remodeling, we use both wild-type and miprin 1 alpha deficient mice. We treat the mice with angiotensin II using mini pump for 4. As shown in the figure A and figure B, measurements of the ratio of heart weight versus body weight indicate that deletion of miprin 1 alpha significantly suppress angiotensin II-induced heart enlargement. Angiotensin II induced the increase in diastolic and systolic left ventricle arterial wall thickness and posterior wall thickness were also significantly reduced in miprin 1 alpha deficient mice, suggesting that deficiency may be inhibit angiotensin II induced the cardiac remodeling. The role of miprin 1 alpha in cardiac structure. We examined myocyte hypertrophy in mouse hearts using WGA staining. As shown in figure C and figure D, we found that significant attenuated angiotensin II-induced and furthermore, the expression of the cardiac hypertrophic biochemical markers such as ANP, BNP, and beta significantly reduced in miprin 1 alpha deficient mice, suggesting that deficiency of miprin 1 alpha may attenuate angiotensin. We also assessed the cardiac fibrosis by series red angiotensin II-induced cardiac fibrosis miprin 1 alpha deficient mice compared with the wild-type mice. Myofibroblasts, the cells essential for cardiac fibrosis, has been shown to express alpha-SMAR and paraostin to enhance synthesis of extracellular matrix, such as collagen 1 and collagen 3. Our Western blot and qPCR analysis shows that deficiency of miprin 1 alpha could significantly reduce angiotensin II-induced fibrosis indicated by the biochemical markers expression, such as alpha-SMAR, paraostin, collagen 1, and collagen 3. This data suggests that angiotensin II-induced cardiac fibrosis were reduced in miprin 1 alpha deficient mice compared with wild-type. Consistently, we also stain the heart section with the different antibody to show the fibrosis. We found that deficiency of miprin 1 alpha could significantly reduce the protein expression of alpha-SMAR, paraostin, collagen 1, and collagen 3, further confirm that angiotensin II-induced cardiac fibrosis were reduced in miprin 1 alpha deficient mice. Consistent with the previous studies, our data show that angiotensin II had no obvious effect on heart function, indicated by EF and FS, ejection fraction and fractional shortening. To further explore the role of miprin 1 alpha in heart failure, we construct a TAC-induced heart failure mouse model. As the figure A and figure B showing that both the shortening were markedly decreased in mice subjected to TAC compared with the Xiao group, while MAPRI-1 alpha deficiency diminished this effect. Deficiency improved cardiac function. In addition, we found that MAPRI-1 alpha deficiency cardiac remodeling assessed by body weight ratio, and the WGA staining showed that reduces the cardiac hypertrophy and the biochemical markers of hypertrophy, including MHC. This data together deficiency inhibited TAC-induced cardiac hypertrophy. To evaluate the role of MAPRI-1 alpha in cardiofibrosis, we found that deficiency of MAPRI-1 alpha could inhibit, indicated by the percentage of fibrosis, also indicated by several biochemical markers of the fibrosis, including alpha-SMAR, peristaltin, collagen 1, and collagen 3. So these data together suggest that MAPRI TAC-induced cardiofibrosis. To further investigate the potential MAPRI-1 alpha blocker in cardiac with the MAPRI-1 alpha inhibitor, Actinonin, which is a small molecule inhibitor, and inhibit the activity of MAPRI-1 alpha by chelating the side cleft. Actinonin inhibit MAPRI constants in the nanomolar range and MAPRI range, indicating that actinonin of MAPRI-1 alpha, but not MAPRI-1 beta. So to investigate the potential for therapeutic application of MAPRI-1 alpha blocker in cardiac remodeling, we treated mice with the MAPRI-1 alpha inhibitor, Actinonin, which is a small molecule inhibitor and inhibit the activity of MAPRI-1 alpha by chelating the catalytic zinc in the active site cleft. Actinonin inhibits MAPRI-1 alpha with the inhibition constants in the nanomolar range and MAPRI beta in the higher micromolar range, indicating actinonin mainly inhibits the activity of MAPRI-1 alpha, not MAPRI-1 beta. So we treat the mice with the actinonin and then use the TAC-induced cardiac remodeling. We found that as the figure A and figure B show that after we treat the mice with actinonin, it can significantly improve the cardiac function indicated by the fraction and fractional. I've shown that the WGA staining demonstrated that actinonin can significantly suppresses the TAC-induced myocyte hypertrophy. Consistently, we show that actinonin could significantly suppresses the TAC-induced fibrosis. This data together suggests that actinonin elevated TAC-induced cardiac dysfunction and remodeling. As we mentioned before, MAPRI-1 alpha as a metallar protease could enhance LPS-induce the production of TNF-alpha and L1 beta. So we are wondering whether MAPRI-1 alpha could contribute cardiac remodeling by regulating inflammation. To test this hypothesis, we first used a MAC3 antibody to stain the mouse heart section, we found that deficiency of myprin-1 alpha significantly decreased the MAP3 positive macrophage, but not CD4 positive T cells and CK positive mast cells of myprin-1 alpha. Decreased angiotensin II induced infiltration of macrophage in heart. Furthermore, we detect whether myprin-1 alpha could mediate the pro-inflammatory cytokine production in mice. We found that deficiency of myprin-1 alpha could significantly reduce angiotensin II IL-6 and IL-1 beta production, but not IL-18, TNF alpha, and MCP1. Further, this result in vitro, we isolated peritoneal knockout mice. As the figure A shows, alpha blocked in knockout mice. When we treat the cell with angiotensin II, we found that deficiency of myprin-1 could block angiotensin II, such as IL-6 and IL-1 beta. Actinonin could also suppress angiotensin II-induced pro-inflammatory cytokine production, such as IL-6 and IL-1 beta. This data together suggests that myprin-1 alpha in macrophage enhance expression of IL-6 and IL-1 beta. So our previous data suggests that myprin-1 alpha expressed in macrophage could mediate secretion of pro-inflammatory cytokines to contribute cardio remodeling. Whether myprin-1 alpha could directly mediate cardiofibroblast fibrosis and cardiomyocyte hypertrophy, then contribute cardio remodeling, were unknown. To test this hypothesis, we first detect the expression of myprin-1 alpha in cardiofibroblast and cardiomyocyte. Myprin-1 alpha is expressed in both cardiomyocyte and cardiofibroblast at an equivalent level to macrophage, which was reported to. So, what's the function of myprin-1 alpha in cardiofibroblast and cardiomyocyte? To evaluate the function, we use the SH myprin-1 alpha cardiomyocyte. As the data shown that adenovirus significantly reduces myprin-1 alpha expression in cardiomyocyte. Then we treat the cell with angiotensin II. As the figure E and figure F shown that after knockdown myprin-1 alpha in cardiomyocyte, significantly reduced angiotensin II induced a myocyte hypertrophy indicated by several biochemical markers such as AMP, BNP, and beta MHC. Similarly, we use the same SH myprin-1 alpha adenovirus to treat the cardiofibroblast. As the figure shown that SH myprin-1 alpha adenovirus significantly reduced the MAP1 alpha expression in cardiofibroblast. And this, after knocking down of myprin-1 alpha, significantly inhibit angiotensin II-induced cardiofibrosis, indicated by the reduced expression of several markers, including alpha-smart periostin, collagen-1, and collagen-3. So this data suggests that deletion of myprin-1 alpha suppressed angiotensin II-induced activation of cardiofibroblast. To further confirm this result, we isolated primary culture cardiomyocyte and cardiofibroblast from wild-type and knockout mice. as the data shown that in the cardiomyocyte, deficiency of myoprin-1 alpha could reduce angiotensin to induce the myocyte area wise, and then could reduce angiotensin to induce the hypertrophy. Similarly, we found that in the primary cardiofibroblast, deficiency of myoprin-1 alpha could reduce its cardiofibroblast activation, indicated by several biochemical markers, such as alpha-SMAR, paraostin, collagen 1, and collagen 3, suggesting that deletion of myoprin-1 alpha inhibit angiotensin to induce activation of primary cardiofibroblast. We are wondering whether we inhibit the myoprin-1 alpha, then whether it can affect the cardiofibroblast activation and myocyte hypertrophy. So we use the myoprin-1 alpha inhibitor, Actinonin, to treat the cells. As the data shown that Actinonin treatment could significantly reduce angiotensin to induce the myocyte fibroblast activation, indicated by several biochemical markers, including hypertrophic markers AMP, BMP, and beta MHC, and several fibrotic markers alpha-SMAR, paraostin, collagen 1, and collagen 3, suggesting that actinonin could inhibit angiotensin to induce the cardiomyocyte hypertrophy and fibroblast activation by inhibiting myoprin-1 alpha activity. So our data suggests that myoprin-1 alpha could contribute cardio remodeling by regulating cardiofibroblast activation, cardiomyocyte hypertrophy, and infiltration of macrophage and pro-inflammatory cytokine production. But what's the mechanism? How myoprin-1 alpha mediate cardio remodeling? We found that myoprin-1 alpha has been demonstrated to activate ARC1-2 signaling pathway by activation GF receptor signaling pathway. And then ARC can translocate into the nuclear to induce the pro-inflammatory cytokine and some fibrotic genes, related genes expression. So we are wondering whether myoprin-1 alpha could contribute cardio remodeling by regulating ARC1-2 activation. To test this hypothesis, we use the SH myoprin-1 alpha adenovirus to treat both cardiomyocyte and cardiofibroblast. As the data shown that after knocking down myoprin-1 alpha in cardiofibroblast and cardiomyocyte could significantly reduce the ARC1-2 activation. Consistently, we found that deficiency of myoprin-1 alpha in peritoneal macrophage could significantly suppress angiotensin to induce ARC1-2 activation. Importantly, we found that deficiency of myoprin-1 alpha to induce ARC1-2 activation. So these data together suggest that myoprin-1 alpha mediated ARC1-2 activation and contribute to cardio remodeling. In summary, our data suggests that myoprin-1 alpha promotes angiotensin to induce cardiac hypertrophy and fibrosis. Myoprin-1 alpha also increases angiotensin to induce the infiltration of macrophage in mouse heart tissue and promotes the expression of pro-inflammatory cytokine IL-6 and IL-1 beta in macrophages. So this project done by two graduate students, Ge Weipeng and Hou Cuiliu, and two instructors, Gao Ran and Hong Mei. So we thank National Scientific Foundation Committee to support our findings. And we thank Key Laboratory of Medical Molecular Biology to support research. And thanks our college to support our group.
Thank you very much, Dr. Wang. That was a fantastic presentation, given under very difficult circumstances with the internet challenges there. So thank you very much. I think a key strength of your study is that it is very comprehensive. I mean, you've used two interventions, the angiotensin II model and the TAC intervention. You've done it in both knockout animals and with the inhibitor, and then you've looked at the different cell types. So really, really impressive study and a very robust data set. So congratulations. I thought I might start off with a question about the three different cell types that you've looked at. So the cardiomyocytes and the fibroblasts and the macrophages. I think in your study, you used a global knockout mouse, which was obviously deleting Meprin 1 alpha from every tissue. So have you considered using a tissue-specific knockout to tease out the relative importance of Meprin 1 in those different cell types?
I'm sorry, the internet is not very good, but what I understanding is that your question is about the specific knockout about Meprin 1 alpha. I'm correct? So yes. Actually, it's a very good question. Yeah, thank you. It's a really good question. Actually, at the beginning, we used the global knockout of Meprin 1 alpha because we don't know what's the function of the Meprin 1 alpha at the beginning. And based on previous studies, we think that maybe the major function of Meprin 1 alpha in cardio remodeling is depend on the macrophage. But surprisingly for us that we detect the expression of, when we detect the expression of Meprin 1, we find that also in both cardiomyocytes fibroblast. So we have the chance to cardiomyocyte the expression of Meprin 1 alpha in cardiofibroblasts and cardiomyocyte, then we can see what function difference between this. Is that possible? Can you hear me?
Yes, thank you. We have just turned off our videos to try and improve the quality of the call, but we got most of that answer. Thank you. So we have a question from someone in the audience who says, thank you very much for a great talk. And MEP1A is a very powerful in cardiac remodeling. And what they want to know is, did you look at mitochondrial function, size, or oxidative phosphorylation changes with MEP1A deficiency?
Yes, actually, it's a good question about the mitochondrial function, because it's very important that the oxidative stress and the mitochondrial function dysfunction can affect the cardio remodeling. We actually, in this work, we did not show any data about the function of Meprin 1 alpha and the mitochondrial function, but we actually trying to see whether it can affect the oxidative stress, because it affects the ERK signaling pathway. So we hypothesize that maybe it can affect the oxidative stress, but so far we don't have the enough evidence to support the function, the Meprin 1 alpha can affect the mitochondrial function.
Thank you.
We actually can, but we actually can hopefully in the future we can show some data in the future.
Someone else has asked about PDE4 inhibitors and whether these can overcome the MEP1 effect. So is there a role for cyclic AMP signaling in all of this?
I'm sorry. PDE4 inhibitor overcome Meprin 1 alpha. We actually did some we actually did some work about the PD4 in vascular disease, but because the PD4 is a specific enzyme can mediate the cyclin AMP signaling, but whether it will have the crosstalk between PD4 signaling pathway and Meprin-1 alpha signaling pathway, we actually don't have the enough data to support that. So so far, I'm not sure whether PD4 inhibitor could mediate Meprin-1 alpha effect.
We also have a question about the small molecule inhibitor that you used. So my understanding was that it chelates the zinc that is required for activity of Meprin-1 alpha. Is anything known more about the specificity? You showed that that it primarily targets Meprin-1 alpha over Meprin-1 beta, but does it target other metalloproteases as well?
Okay. So far, based on the publication or based on the data of the actinin, it's only inhibit the Meprin-1 alpha, but not the other metallar protease like the MMP. At least it does not affect the MMP activation.
And what's its toxicology profile like? Is it well tolerated by the animals? Do you think it's a potential therapeutic that could be translated into large animal models, for example?
Yeah, actually, that's a very important thing. What we also focus on that. Actually, at the beginning, when we tried this inhibitor, we did not see the toxic effect of this inhibitor in the animal. And also, there are some other publications which demonstrated that the actinin can inhibit tumor formation, that mainly inhibit the cell proliferation and cell migration and invasion. So I think so far in our dose that we did not see the toxic effect of the actinin in animal models. We actually check the kidney, the lung, the heart, the intestine. We did not see the significant toxic effect.
And in this study, it was essentially a prevention study because you were co-treating the animals while they also received the angiotensin II infusion or while they were developing pressure overload induced remodeling. So have you done a rescue experiment where you induce cardiac remodeling first and then administer the compound? Does that rescue the phenotype?
It's a really good question, because I think if it's possible, then next time or in the future we will try this. Because at the beginning we wanna try at the beginning when we test the effect of the actinin, we pre-treat this drugs before the angiotensin II or TAC-induced the cardio remodeling. But if, like you said, if we wanna see whether it can reverse the cardio remodeling, we should induce the cardio remodeling first and then treat the mice with this.
I wanted to ask you a little bit more about the mechanism. What's upstream of Meprin-1 that's actually activating it? There was a little bit of discussion in your paper about how Meprin-1 might be functioning by upregulating transcription of some cytokines, and you suggested that it wasn't necessarily an enzymatic mediated process. So I'm just wondering a little bit more about the mechanism, like if is the enzymatic activity of Meprin-1 being activated, or is it acting in some non-canonical way to upregulate cytokine transcription?
What I can say now is that based on our data that we did not see the expression change of microenvironment alpha after the cardiac remodeling. So now, based on our data, we think that microenvironment alpha mediated cardiac remodeling by depend on the activation of this enzyme, but not the expression of this enzyme. So based on the activation of microenvironment alpha, it's depend on the zinc. So it's a zinc-dependent proteinase. So we think zinc maybe play an important role in biological systems as bond. So although the zinc concentration in the nanomolar range in the cardiomyocytes at rest, but it has been reported under stimulation, it can significantly increase the concentration of zinc. So we hypothesize that maybe angiotensin II or other stimulus can increase the activity of microenvironment alpha through regulation of the zinc concentration. But whether the activity of microenvironment alpha increase during the cardiac remodeling or whether it's depend on the concentration of zinc needs to be further investigated.
Mm-hmm. So we have, oh, sorry, sorry, great answer. So we've got time for one or two more questions. So one person is asking, have you looked at this in a model of myocardial infarction, and is it possible to exert similar effects for post-myocardial infarction fibrosis? Oh,
it's a good question about the myocardial infarction fibrosis. Based on we did not have the data to support that. But based on the expression of microenvironment alpha in the cardiomyocyte and cardiofibroblast, we hypothesize that it may also affect the fibrosis post-myocardial infarction. But I think it also depends on whether the myocardial infarction will activate microenvironment alpha activity, or it can mediate the microenvironment alpha expression. That will be the first question. I think if we want to try this or test this hypothesis. So if it can affect the microenvironment alpha activity and or the microenvironment alpha expression, maybe it can play the similar role in the myocardial infarction fibrosis.
Excellent. And one final question just to wrap up this discussion. So what's your gut feeling? Which is the primary cell type that is initiating the pathological cardiac remodeling related to MEPRIN-1 alpha here? Is it the macrophages or the fibroblasts or the cardiomyocytes? You've shown that it's playing a function in all of those cells, but which is the cell type that's really initiating this remodeling?
If I guess, if I suppose, I will prefer maybe the function of the macrophage as the initial effect of the microenvironment alpha in the cardiac remodeling, because I think during this process the function of the macrophage is more, I mean, how to say, it's a little bit earlier than the activation of cardiac fibroblast and cardiac hypertrophy. That's what I hypothesize. But if we want to identify this question, just like you said, if we can do the microenvironment alpha specific knockout, we knock out the microenvironment alpha in macrophage, we can see whether it will affect the cardiofibroblast, hypertrophy, and myocyte, and cardiac hypertrophy and cardiofibrosis, which will provide more evidence to see which effect come first.
Yes. So one final question, although maybe it might just be a comment. So related to the discussion before about PDE activity, someone's made the comment that PDE activity is dependent on zinc concentration, which you've indicated is very important for Mepla 1 alpha activity. So do you think that this could be important?
Sure. We also did actually PDE4 inhibitor in the vascular remodeling, and I believe that if during the process of the cardiac remodeling, if they think the concentration of zinc increase, it may affect several enzymes, and maybe it has the crosstalk between the different signaling pathway and provide the synthetic effect for the cardiac remodeling. I really think it's possible. So if actually we have the PDE4 inhibitor and PDE4 conditional knockout, so we will see whether maybe next project we will see whether Mepla 1 alpha can mediate PDE4 signaling pathway. Thank you.
Fantastic. And one final question before we let you off the hook. So one person has a question about microRNAs, and if you've checked if any microRNAs target Mepla 1 alpha, and which might be able to be targeted alongside use of your inhibitor.
Yes, actually, a great question. If at the beginning we actually want to, if during the cardiac remodeling, the Mepla 1 alpha expression is mediated. I mean, if during the process we can see the expression of Mepla 1 alpha is increased, we may focus on the upstream, how the signaling pathway to mediate the expression of Mepla 1 alpha. But things we did not see the expression of Mepla 1 alpha here, so we did not focus on the effect of microRNA on the Mepla 1 alpha, but whether microRNA could mediate Mepla 1 alpha activity by regulating zinc concentration is very interesting. We don't know whether microRNA could mediate the Mepla 1 alpha.
Fantastic. Thank you so much, Dr. Wang. That was a really, really excellent talk and some great questions coming through. While you were off the call during the middle, we had a bit of a poll, and there are people who have tuned in from all over the world, from Europe and throughout Asia. So it's really been a wonderful session, and congratulations again on your paper. I encourage everyone who's on the call to go and read your paper if they haven't already. It's really great. So thank you very much for joining us.
Thank you very much. Yeah, thank you very much.
Thank you.